First of all, thank you, organizer, for invitation and opportunity to have talk here. So I'm work in Vladimir Katanaev lab, and we have a lot of approaches for studying GNAO1 related disorder, and this approach can be combined in this pipeline, and I'm responsible for creating a Drosophila model. So the GNAO1 protein is very conservative, and you can see, and across animal kingdom, all proteins have 354 amino acids. And it's a very conservative, their identity between Drosophila and human, the identity more than 80%. And it means the most of mutation can be mutated in Drosophila genome. It was surprisingly that exon intron structure of most, it's like in Drosophila and human and in C. elegans, and very similar. So it means that it's a very conservative, evolutionary conservative protein, and it encourages us to make a model in Drosophila. Okay. So first of all, we asked what happens if we change Drosophila GNAO1 to human one. And we use CRISPR-Cas9 technology, substitute the Drosophila. Substitute the Drosophila sequences to human one, but we used two steps. Substituted these clusters of exon and downstream clusters of exon with two steps. And normally we used Drosophila DNA with a small amount of substitution, which only change amino acids, Drosophila amino acids to human. So finally we produced human protein without changes in Drosophila genome. We saved regular regions and most of Drosophila DNA. So when we proved the expression of human protein, you can see here is immunostaining using monoclonal antibodies raised again human protein, and which you can see it's the staining only in the brain, only on humanized fly. So when actually this was published, and we can conclude that humanized protein can recapitulate the endogenous function of Drosophila protein. So next, we decided this is a good model, and we can introduce a different mutation in genome. In this case, we also use CRISPR-Cas editing and also recombinase mediating cassette exchange. It's just because initially, in first step, we substitute these exons for cassette, which contain marker genes and flanked with HTTP sites, which can be used in this cassette exchange, and we exchange after that RFP marker to mutated exons. So we obtained several mutations. Some of them is most common, this and two of these, and it's one of them is most mild mutation. And also we obtained precise control, which these flies also passed all crosses and all editings. So it was also inserted wild DNA in this cassette. So it's important, especially for behavioral tests. And here is, we can see that some benefits which bring the Drosophila, because we successfully obtained this mutation, which before we couldn't obtain in mice. So now it's the best characterized mutation in our lab, and we used it after it was shown in our lab that zinc can restore functionality of G-alpha-O. We have done these experiments in vivo, and we've shown that zinc can restore longevity of mutant flies. It's here. Yes, this is a blue one. It's a mutant flies which were fed on with zinc. And also we determine of locomotor deficiency in adult flies. For this we use a negative geotaxis assay. This assay, when flies being tapped on the bottom of a cylinder, they try to go away. It's normal reaction of flies, and the speed of with speed of passing this red line can be counted. And actually here on this graph you can see that normally there are 10 old females, these control flies. These control flies, I mean mutant with control flies with mutation without supplementation zinc. They show quite low activity, locomotor activity. And here two lines which just go together. It's control flies, just wild type flies without supplementation, and mutation flies, mutated flies with zinc supplementation. And this effect still visible, but it's quite less, on all the flies. So, yeah, and I believe that these in vivo results promote idea to use this zinc for clinical case, and now there is this article where supplementation of zinc give positive results. So there is one obstacles which we meet, which we faced when we work with Drosophila. In general, the phenotype which we monitor in Drosophila is milder than manifestation in a patient. So we decided, and we can't see any epilepsy seizure in Drosophila. So we decided to develop system to see some seizure in flies. And so the idea was to have done some conditional knockout. So, and when we have a wild fly get heterozygotes with a mutation against wild type. If we can destroy wild allele, it should be a stronger phenotype. And if we can do it in a tissue-specific way, so we can have viable flies, because if we do it in a total organism, these flies can't alive. It's lethality. So, using CRISPR-Cas9 system, we modify endogenous G-alpha-O, adding tagged this with GFP. And these flies were viable in homozygotes. So we can say these flies like wild type. So they don't demonstrate strong phenotypes. And it means we have tag which we can use like target to destroy gene. So, and we can use RNA interference. We attack the GFP RNA, GFP pieces of RNA. Yeah. And then as a result to have like hemizygotes mutant. So using specific drivers, and we use two drivers, Repogal and this one, to induce a specific conditional knockout of the GFP alleles in the mutant background. When we obtain these flies, we used vortex assay to induce epileptic seizure. So I'm just showing now. Okay. So it should be a vortex, but I don't here. So it's control. There are no any effects. Now it's shakers experiment. The flies have, most of them have seizures, and now with some time, just wait. I'm not sure. Yes, now they start move, and we can calculate the time and compare the time with different. So it's quite visible effect. I can't. Depend on stock. It can take several minutes when all flies move. Start move normally. So it can be calculated, and we calculated it with two different mutation, and you can see it's statistically clear, and all the flies have more effect, and the different mutation can show also different sensibility to these tests. So, but the most interesting things, we use two drivers, and one destroy wild type protein in glial cells, and another in neuron. And this effect, which I show now, it was with driver which expressed this destroy wild protein in glial cells. That's quite it's quite unexpectedly, and it's need more precise studies. So now we have some plans and characterize other mutation which we obtained with all assays we have. And also we want to introduce a new behavioral assay to assess associative learning, short memory and long-term memory. So for this we now have some special equipment for these which we can test, train flies with mutation and control flies with odor with odor and electroshock them, and they then give them choose between two different odors. It also can be calculated statistically, and it's reflect, I guess, reflect manifestation of mental ability in patient. So, and further plan, we want to use this conditional knockout to using a different central nervous system specific driver to probably we will be able to detect which region responsible for different phenotype, which can reflect different manifestation in patient. So, and I want to say thank you, Vladimir Katanaim, and our member of our lab. So, and also I want to say that with Vladimir Katanaim we launched the Humana Fly facility with the aim to break a gap between clinician and researcher, using and just help them build the fly Drosophila model for different disease and in general in neurological disease. Thank you.